All right, so we are moving into chapter 7, which is all about cells. And chapters like this, they always want you to start with the list of old dead guys that got us where we are, what we'll call the pioneers of cell biology. I always hate this part. So luckily I've read some stuff about these guys and they were all deeply weird people. And so I'll throw in a little bit of that, which you can enjoy or ignore as you see fit. You really just need to write down what's in bold print and underlined about them in their little word bubbles that I'm going to put up, and their names and when they lived. Got it? Bold print, underline, names and dates. So to kick things off, we have that dude, Anton van Leeuwenhoek, who was a lens guy. He was a lens grinder and hung out with lens grinders and was one of the first to publicly sort of figure out that if you stacked enough lenses in the right configuration, you could magnify things so much that you could even see things that were previously invisible, right? Things that were too small for the human eye to normally pick out. And so he did that, and he did what you typically expect somebody to do, you know, look at a piece of his own hair or some of his own blood. But then he got kind of weird. There's lots of very careful notes that he took on looking at his own poop under the microscope, noting that he saw stuff crawling around in it, especially after he ate a big meal that included lots of meat, which of course is because meat, you know, the refrigeration techniques weren't terrific back then, and parasites were rampant, and so he was seeing the parasites from his intestines in that meat. So he is credited with sort of being the first microscopist. And then we have, just in time for Halloween, Robert Hooke. I think his face probably looks like that because you know what a hypochondriac is? Someone who always thinks there's something wrong with them. Oh. Maybe even to the point of making things wrong with them with their big fat brains. Yeah, he was one of those, and the way he self-medicated was by drinking a full glass of mercury, which of course is a liquid metal, every day, which was widely believed to have some medicinal properties, but we know now it's toxic and makes you a little bit crazy, but apparently packs quite a buzz. Yeah. 1635 to 1703. But when he wasn't high on mercury, Hooke was really into microscopes as well. And one of the things he'd like to do was slice living or non formerly living tissue real thin so that he could see through it. And when he did that with cork, probably the cork from a mercury bottle, he could see all these kind of regularly shaped square openings in it. Because cork, if you don't know, comes from the cork tree. It's part of the bark of that tree. And what he was seeing was the empty cell walls of those plant cells. And what they reminded him of was the rooms that the local monks in the local monastery lived in. And of course, monks called those rooms cells. So this is actually the guy that named cells cells. Yeah. Just the underlined stuff and the names and dates. Yep. So all these guys were Europeans, and they were all part of basically the first Western version of a scientific community to exist. And that community was called the Royal Society of London. And basically people would come from all over Europe to the Royal Society of London meetings and present what they had been working on. You know, so Hooke would come and talk about cells. And one of the other guys, and this is a little later, okay, and he kind of built on these guys' work. Rudolf Virchow was a physiologist mainly not into cells. Mainly he was into how living things worked on the inside systemically. You know, you look at the different systems He was kind of a creep in some ways. He invented vivisection, which, if you don't know, is cutting things open while they are alive so that you can see how their inner systems function. He was known to like to bring stray dogs to the Royal Society meetings and vivisect them in front of the other scientists. There's stories of like people in the front row getting spattered with blood from these dogs that he's cutting open. Anyway, and physiology has kind of historically been sort of a bloody science. It's not just him. But he did think about cells as well. And one of the great things that he did was to help discredit the idea that living things could come from non-living things. You've seen one of the experiments that somebody else did about that, the maggots in the meat. People genuinely thought that meat, when it went bad, just turned into those maggots. People thought that in barns, the reason you saw so many mice was that the straw would actually transmutate into the mice, right? And actually, Louis Pasteur, famous for the process of pasteurization, one of his early works was to prove that bacteria don't come from chicken broth, right? Because he sterilized the chicken broth and saw that no bacteria grew. Anyway, Virchow basically was the first to publicly say cells come from other cells and not from anywhere else. There's no way living things can come from non-living things. So at a certain point in these Royal Society meetings, two scientists who hang out together, one's a plant guy, one's an animal guy, named Schleiden and Schwann. I think it's Matthias Schleiden and Theodor Schwann, but I might have them backwards. First of all, they each kind of specifically say, the plant guy says all plants are made of cells. The animal guy says all animals are made of cells. And then together, they sort of put all the stuff that's come before into one overarching theory called the cell theory. By the way, congratulations. Unless third period really knocks it out of the park, you're going to win the test average contest. 87, not too shabby. So the cell theory doesn't sound real basic to you, but keep in mind that it hadn't been that long since people had even discovered that cells existed. The first is Schleiden and Schwann's stuff saying that all living things are made of them. Right? It's one of our big 8 characteristics of life. Second, though, they are not just building blocks. They are not just the unit of structure of the human body. You're not just a glued together mass of cells that just provide structure. They also do stuff. They have jobs, right? Like blood cells, red blood cells carrying oxygen, white blood cells fighting disease, nerve cells conducting electricity. They're the unit of structure, but also the unit of function. So there's your old dead guys in a nutshell. From here on out, for a while now, it's going to be cell parts. We are going to go through all the parts of a cell, their structure, their function, how they work together in certain ways. And let's start on the very outside. Let's start with what does every cell, plant or animal, have around it?
Cell membrane.
A membrane. You might be tempted to say wall, but remember that that animal cells do not have a cell wall. Most other kinds of living things do, but animal cells do not. So, cell parts, here we go. The cell membrane. In an animal cell, it's the outermost layer of the cell. In other cells, it's just inside the cell wall. They've all got one. What's it made of? For a sticker. What's the cell membrane made of? Very nice. Phospholipids. Remember? We mentioned these. We even drew one in our first biochem unit. So phospholipids are the main structural molecule. Not the only thing there, but they're the molecule that gives the cell membrane kind of its odd properties. We drew it like that, if you remember. And I bet you can guess what those tails are made of. Fatty acids. Very good. Yep, so the head is a phosphate, which is a phosphorus and 4 oxygens and a couple other little doohickeys. And the thing about phosphate is it's polar. We talked about how water is polar because the oxygen holds onto the electrons a little more strongly. Well, a phosphate is like that too. It has a sort of a distinctly positive side and a distinctly negative side. And the thing about being polar on Earth is that polar molecules attract other polar molecules. And so the word we use for anything that's polar and attracts water is hydrophilic. Literally means water loving. Of course, there's molecules in our brains that don't love anything, but you get the idea. Then there's those fatty acid tails. And the thing about the fatty acid tails is hydrocarbon has no polarity. There's no difference in charge when it's just carbons saturated with hydrogens. So we call that nonpolar. And nonpolar molecules do not attract water. In fact, they basically repel water. Anybody want to guess the word we use for that? It literally means water fearing. Not hydrophilic, but hydrophobic, yeah. And so that property of phospholipids, that they are kind of split personality, that they have both qualities, water attracting and water repelling, means if you shake a bunch of them up in water, they do something weird. They basically arrange themselves, sort of spontaneously, to have as little contact with water as possible on their hydrophobic parts. And what that ends up looking like is this. Now, this is like a ball and stick model type representation, but you can see aqueous means water. So what they do is they stick their tails together. Their tails are the hydrophobic part, so they stick their tails together, their heads on the outside to form a double layer that is, squeezing water away from the parts that don't want to touch it and sticking those heads in the part that does. That, by the way, is the lazy organic chemistry structural formula of a phospholipid. These tails are the carbon chains. What? No, the part you should probably do though, because yeah, they form this planar bilayer first, but then because this part and this part are the two parts that don't want to touch water, then they actually form a circular or spherical thing. And so that's the part you should draw. You don't have to use quite as many if you don't want, but you need to show that the tails face inward and they form a circle like that. And that structure has a name. And the name is lipid bilayer. And we will call that enough for a Monday. I'm going to pass out Chapter 7 guided reading now, and that will be due on Wednesday.